This program is brought to you by Emory University.
The Emory University Center for AIDS Research is pleased to serve as local Atlanta host for AIDS Vaccine 2010, sponsored by the Global HIV Vaccine Enterprise. As an international leader in HIV/AIDS vaccine research, Emory has more than 50 scientists focused on developing an effective vaccine for this challenging disease. Research in the Emory Center for AIDS Research, the Emory Vaccine Center, and its Hope Clinic, Yerkes National Primate Research Center, and Emory University School of Medicine range from basic research in the laboratory to preclinical studies in animals, to human clinical trials, to a biotech startup.
So Emory has one of 20 NIH-funded centers for AIDS research, and the main purpose of that center is to facilitate and stimulate HIV research on campus. It's an infrastructure grant. It provides developmental awards to young investigators. It provides specialized facilities and expertise to investigators so that they don't have to reinvent the wheel every time they want to do something. As part of that center, we have over 100 investigators involved in different aspects of HIV research. And that's everything from mother-to-child transmission of HIV to clinical therapeutic trials of new drugs for HIV treatment, to HIV clinical trials of HIV vaccines. A large number of the participants in the Merck vaccine trial were enrolled here at Emory. To a more basic aspect of trying to understand how HIV replicates, to preclinical studies on HIV vaccines. And I think, one of the highlights of our center from an HIV vaccine perspective has been Harriet Robinson's what we call DNA MVA vaccine. It's a two-component vaccine using an oligonucleotide vaccine with a vaccinia virus-based vector to deliver proteins, HIV proteins, to participants. That was first tested in animals here at Emory, shown to be successful, and then has been moving into first safety studies in humans and now more efficacy studies, really more, I would say, ability to stimulate the immune response studies, but hopefully onto efficacy studies over the next few years. We've got studies using that same approach to see whether we can stimulate the immune response of already infected individuals and use that as a therapeutic vaccine. So the whole spectrum, really, of moving from animal testing to human testing of HIV vaccines here at Emory. My own lab and that of Cindy Durdine's and Susan Allen's research program is more focused on the process of transmission. And again, as I mentioned earlier, we work with discordant couple cohorts in Rwanda, in Kigali, Rwanda, and Lusaka, Zambia, that we feel will be giving us clues to the nature of the virus that initiates infection in a previously uninfected person, that we can then use to refine our vaccine development programs to really focus on perhaps specific properties that those viruses might have.
The HIV epidemic globally continues to be one of the major public health threats. Despite all the advances that have been done up to now, there's over 1 million persons infected every year by the AIDS virus around the world, most of them in sub-Saharan Africa. But there's an increasing number of infected individuals in other places in the world, like, for example, India, Southeast Asia, and China. Here in the United States, the epidemic continues to be of importance. There are approximately a little bit over 1 million individuals living with HIV in the United States. And CDC estimates that about 55,000 individuals acquired HIV infection every year in this country, which means about one new infection every 9.5 minutes. Atlanta is an important place for the epidemic because the epidemic, while in the United States it started in San Francisco, in California, in New York, it increasingly is an epidemic that is more severe in the South, in the southeastern United States. So clearly we are at what I would call the epicenter of the current HIV epidemic. The HIV epidemic in the US and here in Atlanta in particular affects primarily minorities, African Americans, and poor people. And therefore it's an epidemic that over time has begun to put a much higher burden in those that are less well off.
A centerpiece of the Georgia Research Alliance is to help our universities attract top talent, the best and brightest researchers from around the world. And I think our emphasis on vaccines really would date back to the early 1990s when we collaborated with Emory University to recruit Dr. Rafi Ahmed to Emory. And it was really Rafi's vision and the vision of others to build a world-class research center here at Emory University, the Emory Vaccine Development Center. And since that time, we've helped Rafi build from, I think, just a couple of principal investigators to an enterprise that numbers in the hundreds today.
The goal of my laboratory is to develop an effective AIDS vaccine, and of course it's our dream. We work on 3 different approaches. We work on developing a preventive vaccine that is given to people ahead of infection, and a therapeutic vaccine that is given to people who are already infected. And we are also working on treatment approaches that will reduce the viral burden in HIV infected individuals. Ultimately, we would like to combine the therapeutic vaccine with the treatment approaches to get the best outcome. Working with Harriet Robinson, we developed a vaccine that is a DNA prime MVA boost approach that is currently being tested in humans in US. This vaccine is in phase 2 trials, and my lab is further working to enhance the efficacy of this vaccine by using adjuvants such as GM-CSF and CD40 ligand.
I think the first and primary challenge is that we don't have a perfect animal model for testing candidate vaccines. So we're always using approximations as to what we think is going to be the best model. And then the second is having adequate funds for a focused program. And it takes a lot of money to take a vaccine forward, and our vaccines go through peer review, and there's lots of opinions as to what should go forward. And the further you get with your vaccine, the more expensive it becomes and the larger the decisions are. I think in AIDS vaccine research, just critical findings have been the results of the large clinical trials. And these have shown that antibody alone doesn't work, and that T cells alone don't work, and that antibody plus T cells hold some promise. And I think if my vaccine has distinguished itself at all from the vaccines that are currently in clinical trials, is that we have relied very heavily on our results from our non-human primate challenge studies in the choice of what components we've taken forward.
Our lab is focused mostly on the strains of HIV-1 that circulate in sub-Saharan Africa. So they're a little bit different from the viruses that circulate in the United States. And we've spent quite a lot of effort and time trying to understand how these viruses differ from the viruses that were studied, the first part of the epidemic, here mostly in the United States and Europe. So the way that we do that is we are working with discordant couple cohorts in Lusaka, Zambia, and Kigali, Rwanda, and these are collaborations with Eric Hunter and Susan Allen that are also at Emory. And we're interested in a couple things. One is how the virus gets transmitted from person to person, and so these are heterosexual couples that pretty much live together, and in some cases one partner's infected and the other partner's not. And so one of the goals is to try to prevent transmission, but it's not a perfect system, so sometimes they do transmit the infection, and we would like to know how that happens and how the virus gets from one person to another. We know that there is what we call a genetic bottleneck, so all of the viruses don't get across from one person to another. So we're trying to understand that process and what might be different about the virus that does start an infection and how we can block that with a vaccine or other therapeutic interventions. So that's one of our interests. Another interest really happens after the new infection starts, and the virus starts off really as a single virus, and then when it encounters the immune response, it starts to change itself. So it makes mutations, and in this way it's able to escape from the immune response. And so we're very interested in how it escapes from antibodies, which are part of the humoral immune response. And we study viruses and the newly infected person over time to try to find out how they're responding to the virus and then how the virus escapes that response. Because unfortunately, the immune system really puts up a good battle, a good fight, but most of the time it loses and the virus wins. And so we really want to know at the molecular level how that happens. And the reason that's important for vaccine design is because we don't really know how to make these kinds of antibodies with a vaccine. We don't really understand maybe what the virus looks like in its real state and what the antibodies see. And so what we do in our lab is try to go in great detail and understand what the virus looks like, what the antibodies see on the virus, and then how the virus changes itself to escape.
We've learned from work with vaccines, and we've learned from work with AIDS that the involvement and support and direction of the community is important. With AIDS vaccine trials, it's important to have an understanding of what is known about vaccines and what isn't known, and an understanding of what can be learned through participation in trials. When vaccines are developed, it's extremely important that the community be involved in their implementation. A vaccine is no good if only half the people agree to have it. And we've seen with childhood vaccines the importance of communication with communities. So the time to get the community involved is the very beginning and throughout the process.
The community has been very actively involved in both as advocates and as participants in studies, as well as cheerleaders in the AIDS vaccine work. You see groups all the way from Action Cycling Atlanta that does a significant fundraise to benefit HIV vaccine research here in Atlanta, all the way to African American church leaders who are actively involved in promoting HIV vaccine research as an important thing that their community should be involved with. I think AIDS vaccine research has been probably one of the most scientifically humbling enterprises we've had. At almost every step of the way, we have found that there's been unexpected results, that there has been challenges, and therefore we are nowhere close to where I wish we could be. 10 years ago, I would have loved to see us have by now something that was much more effective than what we currently have. There is, however, recent results from one study suggesting that there may be some protection with a new vaccine. Not a lot of protection, but at least it begins to offer some hope that maybe there's something there that needs to be pursued. But the development of vaccine of an AIDS vaccine has been an incredibly challenging enterprise, and something that really has required a lot more scientific brain power and a lot more imagination that we've been able to accomplish up to now. It really has remained a riddle that we have yet to crack.
The field was quite surprised about a year ago when a very large trial in Thailand, the so-called Thai trial, actually yielded moderately effective protection. Again, a vaccine that was a two-component vaccine, a viral vector prime with a protein boost, the very same protein that failed in the Vaxgen trials, but provided a moderate level of protection, so that in that trial about a 30% decrease in viral HIV acquisition was observed. And I think that's re-energized the field. It's said, I don't think it's taken us away from this realization that we still need to generate more basic science knowledge. But it did suggest that success of an HIV vaccine wasn't impossible. And I think having that possibility of success has been very energizing and has got the field really thinking forward again about, well, what other mechanisms might also be important in the success of an HIV vaccine. And part of it is probably going to be neutralizing antibodies. Part of it is probably going to be an active T-cell immunity. But maybe there are some other components, innate immune components, non-neutralizing antibody components that we've not been focusing on particularly, although certain investigators had, that now I think we're trying to put into one big package that might yield an effective vaccine. So I think it's an exciting time, and I think we're going to see sort of the products of some of the basic science, and we're going to see some of the more in-depth analyses from both the Merck trial and the Thai trial at the conference that I think will be really helpful in moving the field forward over the next few years.